My name is Corrine Kliment. I am an assistant professor and physician scientist at the University of Pittsburgh in our pulmonary allergy, critical care, and sleep medicine division. I have my own lab and I also work clinically. But one of my passions is mentoring kind of the next generation of scientists, physician scientists, and scientists in the lab.
I saw that you described yourself as a mitochondrial biologist and lung repair researcher. So, would you be able to expand on what that means?
Absolutely. So our lab is really interested in several chronic lung diseases, namely some that spur from lung aging. So chronic obstructive pulmonary disease or COPD, and then pulmonary fibrosis or scarring of the lungs. And we know that certain cell types and stem cells within the lung are really important in how the lung repairs and regenerates after being injured. We're particularly interested in how mitochondria and if they become dysfunctional, how that impacts the ability of those epithelial cells within the lung to repair and regenerate, and how this goes awry in diseases of lung aging.
What are some of the ongoing projects in your lab currently?
So we've become really interested in a protein called adenine nucleotide translocase, and it's one of the most abundant mitochondrial proteins that transports ATP out of the mitochondria. And we've found that expression of this protein is low in these diseases of aging, so in COPD and in pulmonary fibrosis. And we believe that that's harmful within the lungs to these epithelial cells that are needed to repair. And so one of our main projects is understanding how that deficiency of this protein alters disease development. But then we're also really interested in how you could harness those proteins therapeutically to actually enhance their function or to be protective within the lung and even the airway epithelium.
So, how does that look like in practice? What techniques or how do you tackle these questions that you have in your lab?
So we're not a one technique lab. We really span the gamut of things. We go all the way from, cell culture and tissue culture with cell lines, but also primary cells directly from either human lung or mouse lung. We use animal models, particularly of mouse with smoke exposure or even fibrotic injury models, and then we go all the way up to human disease. The University of Pittsburgh is a really great place because we do a lot of lung transplants, and so we have access to a lot of human samples through research. And so we try to incorporate those to really understand how is our work translatable and are we asking the right question. And so we go back and forth between all these different models. And then we use a lot of molecular biology techniques, everything from Western blotting to PCR to single cell RNA sequencing and tissue staining. We again, use all of those tools, and people in the lab are trained in all of those tools to be able to then use them to answer the really great questions that they come up with.
How would you describe your research team and your research environment?
So we're a really collaborative research environment. I almost see it as like it's a lab family. Everybody helps each other out. Our lab meetings are really vibrant. Everybody gets to participate and weigh in and share their ideas and their thoughts as projects move forward and evolve. Because I think some of the best ideas definitely do not come from me as the PI. They come from the team, everyone else. So we're not a top-down system. We're definitely kind of a level playing field, even team. And I work with everybody individually and mentor them individually, whether it's a technician in the lab or whether you're a postdoc in the lab. I want to know how you want to grow in the year and what you want to learn, and then how we're going to get you there. And so I work on the individual level with everybody, but then we're really a cohesive team.
Other follow-up question was from the side of the bench to bedside approach that your lab has. I am wondering how your training as a physician scientist has impacted that.
I think I've had a lot of really great mentors along the way that have been physician scientists, and I've learned from them, how you ask good questions and put on a different hat. So, I might be working clinically and I'll experience a patient or a case where I wonder, like, I don't have an answer to some of the questions that come up, or I don't have a therapy for them. And so then that creates more questions in my mind that I bring back to the lab and say, how can we answer this? Do we have the tools to do it? And so I can, I love being able to go back and forth. I think it's one of the most challenging aspects of the career, but it's one of the most rewarding. And I love also being able to share that with students and with trainees to then see when they have that kind of aha moment in
Mm-hmm.
their work. It's really exciting because then you can say, because they can think about, oh gosh, I think we have some patient samples. Let's go look in those. Let's go try to answer the question that way. So um, I think a lot of my prior mentors have really shaped how I what my job is as a physician scientist. But it's a really fun job. I love it.
What have you brought from your mentors into your own mentorship style, and how has it evolved throughout your career?
I think every mentee has different needs, and so I think really keeping clear communication is super important in understanding what their needs are and how those needs change over time. If you're a new student in the lab or new trainee, your mentorship may be more intense at that point in time. You have more needs than if you've been in the lab for 3 or 4 years, where you're a little more independent and you kind of get to fly on your own. So I think that's one thing, is recognizing those differences in the needs of each person. One thing I do with all of my trainees is I have something that's called a compact, which is actually something that lists what are the expectations of the mentee for me as a mentor. What can they expect from me? But then also, what can I expect from them? So it's from everything of like reviewing papers to productivity to all sorts of different things. And I think it just makes it very clear what are everyone's goals, expectations, and I've had that work really well in building mentees training plans and helping to make sure everybody's on the same page.
Yeah. What would you tell someone that is interested in learning more about the work that you do, or even joining your lab?
So I think our lab we're really in a growing phase. We've got a lot of really exciting projects going on, and I think we're looking for really bright minds that want to do creative work and work with a really exciting team. Um, I think we try to share what we're doing on our website to really be able to make sure that everyone else knows what we're also doing, and all the wonderful things that are happening for people within the lab and the great work that they're doing.
So, what is something that you're excited about and looking forward to in the next few years?
As we're trying to kind of build models within the lung to study regeneration and repair, we've started to bring in models like 3D alveolar organoids that are grown from, type 2 lung cells or the stem cells of the lung. So both human as well as mouse organoids, to be able to study therapeutic drug effects, study mechanisms of pathogenesis and exposures. And then we've got some really great collaborators here that we work closely with for things like precision cut lung slices, which are actually, just pieces of human or mouse tissue that you can study the interactions of the cells in the same way, studying therapeutics and things like that. So we've got a couple of really cool models, and then we're hoping to incorporate studying mitochondrial function on top of that. Um, and then I think the final thing that I'm really excited about, that we're sort of just getting started with, is looking at ways to do mitochondrial transfer. So can you
Interesting.
take healthy mitochondria and transfer them to cells with sick mitochondria, which we know happens in all of our chronic lung diseases. So again, we're really excited to have people working on these things and get these things off the ground.
Very cool. Would you be able to share some of the findings that you've had over the years that kind of impulsed this new set of experiments?
Sure. So one of the biggest findings that we've had is that we originally had found using an organism, a model organism called Dictyostelium, which is a little amoeba, a soil amoeba. We found that this adenine nucleotide translocase protein is actually protective. So if it's overexpressed or enhanced, it's protective against cigarette smoke induced injury in the lung. On the same side, we also found that levels of this ANT2 protein and gene are are low in these type 2 cells or regenerative cells of the lung. And so we've been able to show this through organoid models that those type 2 cells that don't have this mitochondrial protein won't regenerate and build and create organoids. And so again, it's implicating this regenerative process that is dysfunctional, and that could be happening. And can we intervene earlier? And so I think it gives us a really nice model of putting in an intervention and seeing directly, will these organoids grow now that we've intervened, related to that protein. We have a second project actually that's looking at this ANT protein, but in macrophages, and showing that they almost undergo this
Mm-hmm.
immunosenescence where they become quiescent and they don't produce, they don't, we don't think that they migrate as well. They don't produce the same kind of cytokines. And so, again, the metabolic state of these cells is super important in how they behave and how they function in their normal environment.
What do you think are some of the main challenges about this type of research?
I think one of the challenges I always have is that there are so many
Mm-hmm.
questions, and just not enough time to answer them all, which is a good problem to have. I think that the lung field in general is a little bit behind as far as therapeutics go, and so I think that's one of our biggest challenges is how do you repair a lung, because you have to repair it earlier in the disease process, because if you catch it too late, the lung's destroyed. And I don't know how yet to build lungs. So I know there are bioengineers that are working on that. But if I knew how to rebuild the lung, then I wouldn't have to be a scientist anymore. We'd be done. But I think the problem, what we're trying to do is, can we intervene
Mm-hmm.
early in the pathogenesis? And I think that's the big challenge, is what's going on in these early stages.
Yeah,
So
definitely. What is something that you wish you knew when you were in the earlier stages of your career or even throughout your training?
the stage where I'm at is, I'm at the stage where I've transitioned from like a career mentored award to my first RO1. So I kind of made that big leap,
Mm-hmm.
right? And I think one thing that I wish I would have learned during training, more than what I really needed, were skills in how to run a team, and not how to run a lab, actually how to run a small business, because the lab is really a small business. Um, we call it, and it's a team. And so I would have wanted to know more about managing people and hiring people and running a small business and all of those things. I just don't think we learn enough
Mm-hmm.
of it. I've had to kind of learn it on the fly, but so far so good.
What is a piece of advice that you can share with graduate students and with MD PhD students specifically about how to be in science in general?
One is to be resourceful, find out always keep that end goal in mind and be working towards that end goal. Sometimes we get really into the nitty gritty of the day to day, but you have to enjoy the process, but you always have to keep that end goal that you're working towards in mind, and then be resourceful in how to get there. And then try to be as innovative as possible, and creative. And if you have an idea, don't ever think that it's too crazy to actually work or to come true. I think follow it, and but again, follow, it's okay to have divergent paths as well. So if, as you're going through your training, you realize, oh, I always thought I wanted to do X, and you realize maybe Y is where you want to go, that's okay. You should go there. And I think your skill sets will still apply. Just find the right mentors to help guide you on the path. But either way, it's a really fun career path, and I always want to encourage people to just stay in it and stay with it.
Thank you. So I want to end on a question about how did you choose what field to work in, both in the medicine side and also the research side?
Sure. So I've done pulmonary research for a long time, kind of since the beginning. I've always liked it, but I did test out during graduate school. I thought I might want to do cardiology, and so I did a cardiology project, and I wanted to just see if it was for me. And all the model systems are a little bit different, and so I just had to test it out. a little bit and see what was right. It was hard for me to decide clinically like which area I wanted to do, but I think I knew in general that I liked the intensive care unit because I like taking care of all of the body systems kind of on steroids. Because these are the sickest patients that we have and things like that. And I think I really enjoyed even kind of the day-to-day mundane things were okay with me. I was okay with those. And as long as you can tolerate those things, then the really fun stuff is what pulls you through as you go forward. So I think it was having mentors that were doing what I wanted to do, and I could say, okay, that's what I want my career to look like, was really important in helping me decide. Pulmonary was also an area where I could use my hands and do procedures without necessarily being a surgeon, which I also considered. And so I think it was for me, it was like a good option that met all the things I was kind of looking for.